there are large data sets of proteins and protein structures. And AI has been pretty good at predicting protein structures. And so that's what kind of gives us optimism that the language of biology, it's a sequence of amino acids or a sequence of nucleotides. Language is a sequence of letters and words. Those things are at some level the same. And so the same kinds of tools can be used to make predictions out of those data sets.
Welcome back to the Smart Biotech Scientist. I'm your host, David Broman, and this is part 2 of our conversation with Jeremy Agr from Triple Bar Bio. In our first segment, we explored how miniaturization is revolutionizing bioprocess development. Now, we'll dive into the exciting intersection of artificial intelligence and biology and discover how high-quality data is the key to unlocking unprecedented possibilities in biologics manufacturing. Let's jump right in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Broman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
And so let's make this very practical. For instance, I'm coming to you and I want to develop this new molecule. What are the high-level steps? Is that always the same, or it really depends on what we want to achieve, or how do you do that?
Yeah, it depends. We tend to work with partners who don't already have something going. We've worked with both. We've worked with partners who have already developed to a certain point and they've reached, a wall with their own capabilities, and we work with them to kind of take them to higher levels. We've done that. We tend to seek out partners where they have a different part of the value chain that they are solving for. So if we want to make a dairy protein, we prefer to work with a dairy company that already knows how to sell and formulate and deal with all kinds of things like regulatory environment for the products. And so they might not have a microbial development team. And so we would start from scratch. So they might come to us and say, we think it would be valuable to make this protein through fermentation because maybe the supply is limited, or any number of reasons why there would be a demand outside of the current production model for that protein. And so we start with a model, like a techno-economic assessment of, for this protein, what scale does it need to be produced at? What kind of efficiency does it need to be produced at to make sense for cost points, really, is what it comes down to. That's the first step. And a protein that sells on the market for a couple dollars a kilo is probably not going to be a great candidate to make with precision fermentation because of the costs involved. It's probably difficult to get down to those costs, but there are many proteins that have higher prices that meet that economic assessment. Then we look at things like what's important about that protein in terms of not just the amino acid sequence, but say, are post-translational modifications really important to the function of that protein? Can those types of modifications be done in a heterologous system, or are they really specific to the mammalian cell that produced it? And then we look at where does that fit in compared to our current, what we've done so far. Maybe we worked on a similar protein, and we take it through this tiered process and ultimately just come up with a plan. Usually we start by taking the gene for that from the natural source and putting it into one of our expression hosts and just seeing not only where we start from, but by applying evolutionary pressure after a few generations, what does the trajectory look like? And then we can see from that what we think that is a reasonable time frame for delivering a product based on that. So that's how it starts.
And then once you have done the development work, you're going to hand it over to, for instance, a CDMO or somebody who's going to produce at a larger scale, I imagine.
Right. Yeah. We're not building any sort of manufacturing capacity ourselves. So we work with either CDMOs, or sometimes our partner has an interest in their own production. If they have a strain that they trusted to perform at scale, they're willing to either build or get access to production space. I think that's part of that. You asked what my controversial opinion is. I think it's related to that. Like there is a limitation in large scale production facilities right now, but there's also a limitation of inputs to that. If we had a spectrum of products that were vetted for their market potential and organisms to produce them at scale, I think it would be pretty straightforward to get the finance building new production facilities. You don't have to use VC money to build a production facility. There's other ways of financing those kinds of projects if the risk has been taken out of the kind of upstream part. That's what our partners work on.
That's amazing. And I think there's a lot of potential there. Now, I want to focus on another aspect because, well, you have the right microsystems in place to screen a lot of different candidates, different molecules, combinations. You're using AI, but this is only as good as the data you're generating. So I just want to focus on the analytics for a second. How did you solve this? Because it's key to have high quality data to make good predictions.
Right. High quality and large amounts of data, right? Like for the large language model approach to AI, like the data needs to be large and like well annotated. And if you look in biology, there aren't that many places where there's available data that is large and well annotated. Interestingly, and I think really optimistically, the places where there are those data sets, like AI has been really effective. There are large data sets of proteins and protein structures, and AI has been pretty good at predicting protein structures. And so that's what kind of gives us optimism that the language of biology, it's a sequence of amino acids or a sequence of nucleotides. Language is a sequence of letters and words. Those things are at some level the same. And so the same kinds of tools can be used to make predictions out of those data sets. The way we solve that problem is we've developed tools that enable us to basically screen for a function. During that screening process, we're able to functionally label genotype information. So the genetics that encode a function with the function itself. And we use that. We've developed some tools around using next-gen sequencing to count different species as they come, species, not in the biological sense, but different types of things as they come through the system. And basically use those counts as a way to associate a function with the genotype. And those become our data set. We have a publication that's in preprint right now. It's just about to be published that we did in collaboration with Google DeepMind, where we basically showed that this is possible using at the level of enzyme development. So using our screening tool to look at the probability that something crosses a gate or not as a measure of function, and then use the counts on either side of that gate to attribute a function to a genotype. And then that becomes a training set. And there are some new tools that need to be developed to scale that to the genome level. We developed the idea of how to do that, and we're working on developing that in-house now to take the concept that we proved out on proteins and scale that to the genome level. And so what we end up with is a data set that has hundreds of thousands or even millions of genomes, the genome sequence, the functional output of that genome sequence, whether that's a production of a molecule or we're applying that in the area in pharmaceutical discovery. That function could also be production of a molecule. It could be a T-cell therapeutic. It could be a function. That genome could encode a function that is like a high potency T-cell for eradicating cancer in a patient, for example. These very complex traits that are really encoded at the genome level.
But these are all high throughput methods, right? And non-invasive because you don't have a lot of material available.
That's right. And the miniaturization helps there, where you can take the material that you generate. Doesn't have to be hundreds of liters of material to do a screen. It all happens in kind of the palm of your hand. And so we can generate these large data sets, orders of magnitude faster and cheaper than you could using the state of the art would be like a liquid handling robot, maybe a high density well plates of being moved around by robotic arms. But those can really only, even in very well funded labs, so we're talking thousands of tests per day, maybe. Right, that you could do.
You have a limitation there.
Yeah, that's right. And when you start to add up a mil, 100 microliters across these tens of thousands of tests, it's a lot of material. The assay components are expensive. It's a lot of plastic. There's a big footprint to doing that kind of testing and really like a cost limitation. So there's been some of the more public companies that work with traditional robotics have published that it would cost them $10 to $100 to get a single genotype sequence from discovery of an improved variant to sequencing that variant might cost $100 per genotype, per winning genotype. Try to scale that to 1 million sequences, and you're talking about like $100 million to look at to make a 1 million member data set. And 1 million genome data set is still pretty small compared to like language models that like ChatGPT is, we're talking like trillions of tokens for like a GPT-4 scale data set. And so that cost for going from I've found the winning data set. I've labeled the function of that data set. Could be a winner or a loser, but I have the functional label of that data point. And the genome sequence has to be way less than $1. And the only way that's going to happen is with miniaturization. And so those are the tools we're developing now is like, how do you get the per data point cost down to just pennies?
You have made a big step ahead. You're able to test a whole lot of different samples at a small volume at a fraction of a cost. I mean, that's already a huge step because what I've seen in my career, oftentimes the availability of data was the limitation, and to make something meaningful out, and especially using AI, because the data sets are just too small to use AI effectively. So I think that's already a big step. So now I'm curious because this is today, we're already able to do such amazing things. Like, where do you see this going now in the next couple of years or even 5 years?
I was saying before, I think that the tools of AI, like the algorithms that have been developed for natural language and where large companies are investing, billions of dollars to do this development of these algorithms, those are progressing, but they start to reach a plateau or it becomes a lot more commoditized, which is great, right? It becomes more available for these applications like biotechnology. So I think that as those more general tools continue to develop, then what the frontier is really the data sets. You said it. We talked about it. That's what's missing. As we start right now, I would say the current state of the art is we're pretty good at predicting the shape of a protein, like with AI. Not every protein, but we're pretty good at that. We're not so good at predicting like interactions. We're not so good at predicting function unless there's been a specific data set for that. So where I see it going is like with the right data, and I think we're going to be a big part of that data kind of revolution, is once we have the functional data associated with the coding information, we start to be able to predict function. So predict not just how an antibody binds to a target, but how it actually, say, agonizes a receptor, or predicting not just how a T cell receptor binds to a cancer antigen on the surface of a cell, but how that functionally activates the T cell to kill that cell. Getting to functional design. I think that's an important frontier at the kind of protein level. And then right now at the genome level, it's very academic. There are a few tens of models that have been developed at the kind of genome language model level. There's very little data, very little diverse data on genomes. And so then feeding that engine with data, now we start to be able to predict genome function. And that's a huge frontier that's really, people are just starting to think about it, is like, running it forward, what if you could design a genome to have a particular function in a computer like that? If we have enough data, I think we can do that. Working backwards, what if we can see the output of a cell, and what if we could diagnose what that genome is? So as a way to like understand disease better by running that kind of model backwards or running it forward in a biotechnological way to make something better. And I think that right now there's not even close to enough data to do that. But I think that by developing tools like we're developing, and then at some level, those different models converge. You have a genome model, and then there's a protein model within that as well. So you can understand regulatory and other kind of important things about the genome, and then also like the function of those things through the protein part of the model as well. And so I think that convergence, a few years away from that still, but I look at it as it's a model that kind of predicts life. Nothing short of that. I think that what's missing right now are not so much the algorithms, but more the data itself.
Wow, that's amazing. I mean, once we are able to unlock all the full potential, it would just enable us to make so many new discoveries and experience breakthroughs. So actually, we should probably have another discussion about that in a couple of years and see what will happen in the next time. Yes, this has been great, Jeremy. What a fascinating conversation. What is the most important takeaway you want our listeners to walk away with from our conversation?
I think that the data things we were talking about before is really important. I think that we can all get really excited about the prospects of AI for biology, but without amazing data sets, the reach will be limited. And so I think I want people to focus on large amounts of data. How do I get large amounts of data and apply that to the problem?
Yeah, definitely. We need large amount of data set, and I would add high quality data sets. I think then we can use the full potential of AI. Yes, absolutely. Well, thank you so much, Jeremy, for sharing all your insights, your passion. Where can people connect with you?
Yeah, so triplebar.com is probably the best way to connect with us. Triple bar is in 3 bars, the mathematical symbol for identity. That's us.
Fantastic. I will leave the link in the show notes, so please reach out to Jeremy. And thank you so much once again, Jeremy, for being on the show today.
Thank you. It's been really fun. Enjoyed the conversation.
And that concludes our fascinating discussion with Jeremy Agr. His insights into the future of AI-driven bioprocess development and the critical role of high-quality datasets are eye-opening. And before we go, are you looking to accelerate your biologics development while keeping costs in check? Develop biologics better, faster, at a fraction of the cost with our fractional CTO services. This is of interest. Book a free call with me and use the link in the show notes to do so. Thank you for listening to the Smart Biotech Scientist. Don't forget to subscribe and join us next time as we continue to explore the frontiers of bioprocess development and biologics manufacturing. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.